Our next presenter is from IDT Biologika, and they are also a CDMO Leadership Award winner. She is Dr. Nanette Schlatermund. She is a senior expert for business development. Welcome, Nanette.
Hello. Thank you very much for giving me the opportunity to talk to you. So first of all, you already see our poll question. Would be very great if you invest a second to read this and answer it during my presentation. Okay, so I would like to give you a capacity update. It fits to this topic about gene therapy from process development to commercial manufacturing. First of all, I want to introduce what IDT Biologika does at all. So we are a contract development and manufacturing organization, focus on gene therapy and immune therapeutics, and we do have a long history in vaccines. We also do fill and finish. We fill and finish products we manufacture in-house for our customers, but also do filling for other sterile injectables. So the goal is really for us to develop high quality manufacturing processes in order to make it possible for our clients to go with their therapeutic and prophylactic products to the market. And some of them might impact just some 100 people, and others are really important, like global vaccine supply against infection diseases. So we also offer end-to-end services, and because this is always a question, what does it end? Where does it start and where does it end? So for IDT, this includes process development, drug substance manufacturing, the drug product manufacturing, labeling, and also secondary packaging of the product in vials or syringes, and a lot of quality control and analytics, which go along with all of the processes I described up front. Where are we located? So right now I'm in Europe, I'm in Italy, but IDT headquarters is in Dessau. Dessau is a pretty small city in Germany, so we are about 1 hour away from Berlin, so the German capital. And in Dessau, we really have our big facility, so it's all this administration, but also the manufacturing side. So we do manufacture everything in GMP quality there. And do also the fill finish, and I will share more details later. The IDT Biologika also has a site in Rockville in the US, so it's in Maryland. We learned already. We do support service for our US customers there, especially in process development. And process development is a big topic at IDT, especially when we talk about gene therapies, especially AAVs. So we also have in Germany, close to our headquarters, facility in Magdeburg. It's a small town with a university where we do a lot of process development and also pre-GMP manufacturing. Some numbers I would like to share. I already mentioned the sites in Germany and in the US. IDT has a very long history. We are more than 100 years on the market. We really started with vaccines for animals. But today, as I mentioned, we are a CDMO and only manufacturing products for human use. We do GMP manufacturing since more than 30 years. The employee number is about 1,600. Most of them are located in Germany, in Dessau, because manufacturing takes a lot of people. And we invested more than 500 million euros during the last decades. This includes new buildings, new filling lines, and especially new clean rooms, of course. We are approved by EMA and FDA, and also for Anvisa, because we do have some products which are also provided commercial products to Brazil. A little bit about our ownership. So we used to be owned by Clocker Holding only, and since October this year, so it's still kind of new, SK Bioscience decided to invest in IDT Biologika. Now I want to shift the topic and make it a little bit more precise. What do we offer in terms of gene therapy? So as an example, I will pick AAV. So just a brief idea how AAVs are manufactured. I guess most of you know already, but I would like to present this here because this is the strategy IDT also uses. So we do turbo plasmid transfection, which means that all relevant genes are separated in 3 different plasmids. And in addition, you need a cell line which does express the E1 protein. This idea of manufacturing is very common because it has a good safety profile. Because we have at the end in the final product a really low viral genome content. And it's easy to scale pretty much. I mean, you don't change a lot, it's just upscaling, but I will come to some of the non-that beneficial product details of AAV as well. And this possibility to work with the turbo plasmid also maximize the space of transgene which is available in the AAVs. So when we talk about AAV, what is really important and what are the challenges? So let me start in the upper right corner. So right now the market is pretty competitive. Sorry. So for some of the rare diseases, some of the sicknesses, there are a lot of clinical trials already in the market. So in order to be one of the first, or better be the first, you really have to be as quick as possible. And that's one point IDT tries to address because we have an AAV manufacturing platform. It is still that the most AAV therapies target rare diseases. So often you need only small amounts of products at the end. And so the manufacturing costs of each treatment is pretty high. So you need to be fast, but the cost per treatment is high. These are 2 points you need to target and address in order to be successful at the end and really bring a product to market. And it still is reality that AAV or in general gene therapies are not that established as monoclonal antibodies. So the EMA and FDA, they still learn a little bit and keep asking for more and extended quality attributes. This is also something, the analytical part, you should not underestimate. And yeah, I mentioned already the costs per treatment. So especially because you need a lot of plasmid and transfection reagents every time you manufacture, this might increase or does definitely increase the costs. So yeah, how does IDT try to target this and make it as cheap and possible and fast as possible for our clients is that we established a platform. So what you see here is our upstream process on the left hand and our downstream. So we really start with a HEC293 cell. That's our established cell line. It's possible to handle this with as an adherent cell or also in suspension. Then we use fat batch reactors or stir tank reactors, do the transfection with a plasmid, and of course grow the virus and do the harvest. Especially in the downstream process, I think there are a lot of things you can improve or IDT did improve. So we decided to go with a 2-step clarification step. We introduced affinity chromatography and also ion exchange. And at the end, for purity and formulation, we also do a TFF. And last step, of course, is always the filling into most of the time vials. This is also something we offer. Especially for AAVs, it's important to track the full empty ratio. So these analytics goes along with our downstream process. I mentioned analytics already twice, and as you can see, we do have a lot of, or there are a lot of requirements for analytic testing. We are at IDT very proud that we do have a big group who does analytic development and also the routine analytic testing. So they are in-house and they are between development and routine analytic up to 120 people doing nothing else. We do all of this always in GMP quality. And I would like to just pick some of these examples. So what we do is the mycoplasma detection using qPCR, and especially the genome titer determination, we use digital PCR. And one major point is also the adventitious virus testing. There we established a next generation sequencing method, which is GMP compliant. We invested a lot of time and efforts and know-how in this, and we are very successful with this. So yeah, please check out for more details. Let us know if you need more. We have white papers for this, for example. Okay, now's the question, when can a potential customer enter into this AAV platform or into IDT services? So some of our clients really just have the idea. So they have the sequence, they have the plasmid. Some of them don't even have the plasmid in GMP quality. So we also partner with other companies who do plasmid optimization for strategic design. We do not manufacture plasmids in-house, so this is also something we kind of buy from a third supplier. Sometimes it's a customer who brings their plasmids, and sometimes we have qualified supplier for this. As I mentioned already, we work with our HEC293 cells. First of all, we would do small-scale feasibility studies, and then, if it's successful, directly go into clinical trial, phase 1 material manufacturing. And as I mentioned already, this could be upscaled easily. As an alternative, because what I just showed was like really early stage, so these are customers who typically startups, sometimes even universities. We also are able and open to onboard customers who already successfully performed clinical phase 1 or even 2. So these are typically more bigger, advanced pharma companies who are already successful. Sometimes they just don't have the capacity in-house, and that's why they want to outsource it. So in this case, we would also support clinical trial material for phase 3, bigger scale, do PPQ batches, and at the end, also the commercial manufacturing, as I mentioned, including secondary packaging, all the quality attributes, QA, QC, and the QP release at the end for Europe and the US, and also for Brazil, wherever you need it. This is a kind of overview about some technical things we do. We put some small pictures, I know, but it's just an idea about our platform development. So we do also do cell line optimization in case the customer already brings their own cell line. Sometimes it's good, but it could be better. So we do some kind of optimization there. We established already a strategic collaboration, as I mentioned, for maybe plasmid improvements. And then we go to the process development. So we really like to do small-scale feasibility studies because it just doesn't cost so much and it's very reliable, and we have a small-scale model which is very supportive. And as I mentioned, the analytical development always goes with it. From the size, what we are able to support in process development, so it's up to 40 liters, or with a fast-backed reactor up to 4 square meters. When we talk about clinical trial, we are still open for fixed bed bioreactors or steel tank reactors. And because we are already so long in GMP manufacturing, we have a lot of staff which are experienced in CTM manufacturing. We do have automated filling capacities, and I already mentioned the QP release. And in commercial scale, so hopefully a lot of gene therapies will be in commercial scale soon, we offer the complete SOPs, master batch records. A lot of them are already available, so it just needs some adaptation, and the QP release at the end. I know it's not really on the focus for AAVs, but we could do it up to 2,000 liters if necessary. I mentioned you have the product, so now we manufactured your gene therapy, but how do you get it to the patient? So you need some filling. This is an overview about filling capacities in general in IDT. So you see these lines, 1, 2, 3, 4, named like this. They are in several buildings and they meet different requirements. So a lot of them are for commercial products, but we also have some of them which are more suitable, more flexible for clinical trial material. So I would like to pick as an example just two of them. So the first is the line number 2. This is really made for commercial big scale. Of course, we use it for vaccines because you can fill up to 500,000 2R vials in one batch. There are free capacities as well if somebody needs them. But here on gene therapies, we would focus much more on line number 4. So line number 4 is also FDA and EMA approved. We have some commercial projects running on this line. Most of the time we use it for 2R, and we have some free slots for manufacturing or for filling 2R vials also on this line. There's a Leo capacity available up to 3,000 vials. Yeah, maybe not the hottest topic. Some people keep asking about syringes. Honestly, right now, there's no free capacity on our syringe filling lines. And the next slide is an overview what IDT in general fills. So our facility is able to handle everything until biosafety level 2. We even had some projects in biosafety level 3 in the past, but that's not possible right now. We handle live attenuated vaccines, inactivated vaccines, recombinant vector vaccines, conjugated vaccines, and of course, any kind of viral vectors. So it's not limited to AAV. I just picked this as an example. We also can do filling for enzymes, any kind of recombinant proteins, antibodies, biosimilars, and also other modalities like oligonucleotides and mRNAs. And I'm very happy that, Perry, you already explained the CDMO awards, because the picture you see on the left side here, it was taken at the DCAT, where my colleagues received this year 6 CDMA awards. So we have a long history with this, and we are always very proud if we get one, and even more than one. It's always a success to our team, and that's why it's a big party to celebrate this. I would like to point out one topic which becomes more and more important to the industry, I would say. It's sustainability. So last year, so in 2023, IDT finished the first IDT sustainability report. It's available for download on the IDT homepage as well. We really try to, first of all, learn about our energy consumption, diversity, and also the supply chain, how much energy and regulatory requirements do we meet there. So we support the UN Global Compact. And I don't know if EcoVadis is known by all of you, probably not. So this is an independent platform where you can answer a lot of questions. You have to prove a lot of things you do, like respect human rights, but also about energy consumption, as I already mentioned. And so it's an open platform. You can participate there. The company who uploads the information does pay for it, but it's available for everybody, actually. You just have to register, and then you can see what IDT does. And in Europe, it's getting more and more common to use this as a compare from different CDMOs. So the customer, especially a big pharma, already try to ask for this. Do you already have an EcoVadis ranking? And so we are very happy that we received the gold medal this year. So it's brand new. And with this, actually, I would like to say thank you. And I think I made it on time.
You did. Thank you, Nanette. Great presentation. Excellent. Excellent job. So you mentioned the competitive environment of AAVs and the the need for speed very quickly, maybe be the first or among the first to commercialize. So I'm going to offer, you said you had capacity at phase 3 in support of clinical trials. So I'm going to use that. So if a drug sponsor is anxious, they're very interested in getting to clinical trial, and they're thinking about transferring their project to IDT. How do you do tech transfer for speed and in getting that moving quickly?
Yeah, so we do have a really well-trained technical people who are expert in the technology, in analytics or whatever. So first of all, they of course need to see all the information the customer wants to supply. So we do have a tech transfer database for this kind. So it's already data protected, of course, because nobody wants to share data in an unprotected environment. And we do have project manager, which are have a long time at IDT, so they know exactly what is required at IDT, so they can ask the right question. And usually this is really a very intense interaction between our customers, our project manager and technical experts in order to generate all the know-how we need in order to manufacture the customer's project as soon as possible.
Okay, great. That's awesome. We're very short on time. You said you invested $500 million or 500 million euros in your history. What kind of plans for investment do you have for the future?
There will be more investment in maybe more different cell lines to be more flexible. And also in filling capacities, because especially for later stage projects, we see high demand, more demand. And because we do have already a lot of projects on our filling line, might be need for more. Yeah.
Okay, great. Great presentation, Nanette. Thank you for being with us. Dr. Nanette Schlatermund from IDT Biologika, and congratulations again on the CDMO Leadership Award.
Thank you. Enjoy.